Hi there, welcome to this video. We're going to be talking about lipids. So, lipids include all types of fats and oils, and basically, the difference is that an oil is a liquid and a fat refers to a solid. Okay, so we're now going to call them lipids, this whole group of molecules. It also includes molecules that are very similar called cholesterol and hormones. Hormones and cholesterol have a very similar structure, but it's very different to the other lipids that we need to know the structure of. So, first of all, we're going to have a look at a lipid molecule called a triglyceride. So a triglyceride, if we break down the word, starts with tri, and we know that tri means three. Second part of the word is glyceride. Now, it's because it contains a molecule called glycerol. Okay, so it contains three something and glycerol, and the three something that it's made of is three fatty acids plus glycerol. Now, the simplest way to draw this is to draw the glycerol on the side like this, and to draw the fatty acids as zigzags coming out here as your three fatty acids. So that molecule is a triglyceride. Now, they might try and push you and say, oh, what's a diglyceride? Di means two, so it would just be like that. What's a monoglyceride? Mono means one, so it would just be like that. Now, these fatty acids are joined onto the glycerol at these 3 points here with what we call ester bonds. And you need to know the structure of glycerol and the structure of the fatty acids and be able to explain how those ester bonds are made. So first of all, the structure of the glycerol is 3 carbons. Off to the right, we've got 3 hydroxyl groups, OH. That means it's an alcohol molecule. Alcohol molecules always have these OH groups. Carbons always have to have 4 bonds. And on the other end of those bonds, we just fill it up with hydrogen. So that molecule there is called glycerol, and you need to know that structure. Then, if we were to draw a triglyceride, we need to add 3 fatty acids. I'm actually only going to show you how to do a monoglyceride because I just want to do one. But the structure of a fatty acid is on one end, it's got a carboxylic acid. Now, a carboxylic acid is the same as what we had on an amino acid. It was the acid part, wasn't it? It's COOH. Well, now I'm going to draw it back to front compared to how we had it on the amino acid. So I'm going to draw it off coming off to the left. So C double bond O, single bond OH, COOH. And it's also made of a hydrocarbon. Now, a hydrocarbon is a molecule that's made of only hydrogen and carbon. Now, it doesn't actually matter for the sake of this how many carbons you draw. Each different type of lipid would have a different type of fatty acid with a different number of carbons. But for now, just draw a series of carbons, however many, with hydrogens all around. Remember, each carbon needs 4 bonds. So that's our fatty acid and that's our glycerol. Then you need to know how does it join together. So it's a condensation reaction, which means it's going to remove a molecule of water. So it's a condensation reaction, which is how things join together, and it's going to release a molecule of water. So we can put plus H2O over here. So if we rub out those molecules or atoms that have been used to make the water, we're still left with an oxygen here. And it's really important you remember that because lots of people, when they're asked to draw this, miss out that oxygen. And then the bond goes from the oxygen over to the C, and it's called an ester bond. And it's formed in a condensation reaction because it produced a molecule of water. If you were to add 3 of those on, you'd then have a triglyceride. These little bit more about these fatty acids. Now, these fatty acids can be called saturated or unsaturated. Now, if they're saturated, that means they've got the maximum number of carbons. They cannot hold any more. Unsaturated actually have space for more carbons. They are not completely full. So, for example, a saturated fatty acid would have its carboxylic acid group at one end, and then it would have its carbons, and each carbon would have 4 bonds, and on the end of each bond would be a hydrogen, and it's saturated. It's holding the maximum number of hydrogens that it can, and it's got all single bonds. The molecule is roughly straight. Now let's look at the unsaturated. So it's still going to have the carboxylic acid at the end, but this time one or more of these carbon to carbons are going to have a double bond. Okay, and where we've got a double bond, I'm just going to redraw it because the double bond actually kind of goes off at an angle, causes a kink in the molecule, so it would go off that way then after that. Now each carbon needs 4 bonds. So this carbon here has got 2 in the double bond, 3, 4. This one's only got 2, so I need to add 2 more and put the hydrogens on. This one here has actually already got 3, 1, 2, 3. So it's got room for a hydrogen just there, but it has not got room for a hydrogen down the bottom there. So potentially there's a space for a hydrogen there, but we can't add it because of that double bond. It can't form another bond to join on to another hydrogen. Same here, this carbon's got 1, 2, 3, so we can draw one more, but it's effectively missing the one from just there. This carbon's got 2 bonds, so it can have 2 more, and this one needs 3 more, one at the bottom. Okay, so you can see that it's got at least one double bond. Now, the double bonds that you've got here to the oxygen does not count. It means at least one double bond between carbons specifically. Okay. And as a result, it's got a bend in it, and we call that a kink. Okay, so it's got a bend in it, it's got a kink in it. So let's have a look a little bit more about the saturated and unsaturated properties. Now, let's have a look at the words that we can use for unsaturated. You might have heard of the word monounsaturated, or you might have heard of the word polyunsaturated. So think about these prefixes. So if it's monounsaturated, it means it's got one double bond, so one kink between the carbons. And if it's polyunsaturated, it means it's got lots of double bonds. So let's have a look at the properties of them a little bit more. So the saturated ones form straight chains. This means that they can line up against each other. So if I was to draw them, they're basically straight molecules, and I can pack them really close together. Okay. Now what then happens is that they form attractions between the chains. Okay, so we get some kind of attractions between the chains, which provide some strength. So attractions between hydrocarbons, for HCs. Okay, you can see that they're straight just there. They can line up neatly. Yeah, you can get lots of them in a small space like that. And you need a lot of energy to overcome these attractions that forms between them. And this gives them a high melting point. This gives them their property. So they have a high melting point, which means that they are usually solid at room temperature. So something like butter is a saturated fat. Okay. You could have something like wax as well, candle wax. That would be a lipid that's solid at room temperature. So you need lots of energy to overcome these, and that's why you need lots of heat to break them apart to turn them into a liquid. Unsaturated fats are not straight, they have kinks in them. Okay, they are not straight, they have kinks in them. So they can't line up nice and neatly like that because of the double bonds. And their shape means that they push away from each other. So if you try to line them up, yeah, they push away from each other. They will not line up neatly like that. Okay, so the attractions that try to bond between them, you can get some attractions, but you cannot get as many attractions between them, and therefore the attraction that's holding them together is not as strong. So you've got less attractions between the hydrocarbons, and therefore you need less energy to overcome them. Just realised I never put the M on that one. Overcome them. Okay, and therefore they tend to be a liquid at room temperature. So something like an oil. Yeah, because these are really easily overcome. They're really easily separated out. These really easily form the molecular arrangement of a liquid where the molecules can move around relative to one another. So they've got quite a low melting point. They're liquid at room temperature, and this is because of the kinks and therefore less interactions between them. And it forms basically oils. Now, another type of lipid that we need to know is a phospholipid. So let's do a recap of what we've done so far. So far we've looked at triglycerides. Now, the job of a triglyceride is basically energy storage. Okay, and they're made of glycerol and 3 fatty acids joined with ester bonds, and these fatty acids can be either saturated or unsaturated. The next type of lipid that we're going to look at is something called a phospholipid. Now, a phospholipid is very similar to a triglyceride, but with a bit of a difference. So the structure looks something like this. So hopefully some of this is familiar. So this here is the glycerol, same as we had in the triglyceride, glycerol. Then in the triglyceride, we had 3 fatty acids. Can you see what we've got in the phospholipid? We've got 2 fatty acids, so similar, but a little bit different. And then here we've got a phosphate group. So this is totally different. The triglyceride did not have a phosphate group like the phospholipid has. So that's our difference. So they have a slightly different structure and they also have a different job, their different function. They're not used for energy storage. The place where you find phospholipids is in cell membranes. And in a cell membrane, these phospholipids form phospholipid bilayers. So you might remember that in a cell membrane, we've got 2 layers of these phospholipids. So these here are what we call the phosphate head. So that's kind of this structure here. And then we've got the 2 fatty acid chains, which we draw like this. So we usually simplify a phospholipid to this structure. And we say that it's got a hydrophilic head and a hydrophobic tail. So the head is attracted to water, hydrophilic, the tails repel water, hydrophobic. So we've got a hydrophilic head, which is the phosphate part, and we've got hydrophobic, which means repels water, tail, which is the 2 hydrocarbon chains sticking out. Okay, so we do a little bit more about that when we do the membranes. Let's just make sure you could recognise a more detailed diagram of that. So the phospholipid, we've done the full structure of a triglyceride. The phospholipid is similar. So remember that the glycerol was carbons down the middle, OH out the side, and then hydrogens all around the rest of the molecule. Then we're going to have 2 fatty acids joined on. So remember your fatty acids. Along these lines. Remember, it doesn't matter really how long the carbon chain is. Now it only needs 2 rather than 3. Okay, so draw one more of these. So I've got 2 fatty acids, and then in a moment I'm going to draw the phosphate head. You don't need to be able to draw the whole phosphate head, but you do need to be able to recognise it. Now the key thing about the phosphate head is the P. We're looking for the P, that's the telltale sign. So remember how these join on with ester bonds, so it's going to make molecules of water. So we can say +2 molecules of water, like that. We take those molecules out that are involved in making the water. Sorry, that oxygen stays. And then the ester bond is just like that. Okay, and then we need to add a phosphate group on. So the phosphate group fixes onto this third carbon. And what you'll see is, you don't need to be able to know how it forms on, but from the carbon here, we then go to an oxygen. Sorry, it's confusing. So this has been switched around actually. So this one is really this one should be, you'd think, the OH. I don't know where my oxygen went there. The OH there. Now actually what happens, for anyone that wants the detail, is the way they draw it, they just kind of switch them around. So normally you'd say the H is there, the OH is there. Now the way we draw it in a phospholipid, what we say is that actually those two swap. So we've got the hydrogen on this side and we've got the OH here. And then from the phosphate group that joins on, part of forming that reaction gets rid of the oxygen, so then we've still got the O going off to the bond. It's the same as that structure really on there, just we're just drawing it out the other side. So from the O, it goes to a P, and then around the P, we've got the rest of the structure, something like that. So don't worry about being able to draw the phosphate group, but look for a little structure that's got a P in the middle, P for phosphate. So that's your phospholipid. So if you see a structure that looks a bit like glycerol in the middle with 2 fatty acids out the chain and something with a P, that will be the phosphate group sticking out the side. That's your phospholipid. Now, one more thing, I just want to show you how they simplify drawing lipids sometimes. So sometimes when they're drawing, for example, the triglyceride, they might draw the O coming out here, then it goes to the C with the O up there, okay, which is like the full version. But then often, instead of drawing the whole hydrocarbon, they might just draw it as a zigzag, and that means hydrocarbon. Now, each corner on that zigzag represents a carbon. So each corner is a carbon, okay? It's just that they're simplifying it, not drawing it out. You're expected to know that that is a carbon chain there. So that carbon chain would look something like that. And then you're expected to know that every carbon would have 4 bonds, and that on the end of each of those bonds would be a hydrogen. So it's the same as the full version that we've been drawing, okay? But they're trying to help. They're trying to make it simpler so you haven't got so many letters all over the page, okay? But sometimes it can be a bit confusing. So really that zigzag represents that, but they'll often just draw it as a zigzag like that. Okay, so just be ready for that simplification. If they were drawing a saturated hydrocarbon, they draw it like that. If they were drawing one that's unsaturated, where the bit's unsaturated, they draw a double bond like that, and you will see the kink. Okay, the kink will be evident. There you go. Where the double bond is, you can see the kink and you can see the change in direction. So that shows you that that's unsaturated and that's saturated, because that's just a straight chain. I know it's a zigzag, but it's straight, whereas that's got a kink in it. It's unsaturated. Okay, and that brings us to the end of lipids.